Our next speaker is Mariano Carossino. Mariano received a DVM degree from the School of Veterinary Medicine, Universidad del Salvador, Buenos Aires, Argentina in 2012. During the last year of his program, he participated in a short training in diagnostic pathology at the OIE Reference Laboratory for bovine spongiform encephalopathy at the Institute of Pathobiology, National Institute for Agriculture Technology in Buenos Aires. Shortly after graduation, he performed intensive training in virology and laboratory diagnostics at the Equine Virology Unit, Institute of Virology at the same institution, and became a co-instructor of the infectious diseases course at the School of Veterinary Medicine. Mariano joined Dr. Balisarria's lab as a Fulbright Scholar in 2013 in order to pursue a PhD in the area of virology. Mariano.
Thanks. Well, as you all know, horses are world travelers just as we are, and they can travel through the Atlantic and from northern to southern hemisphere in just a matter of hours. And this usually leads to think that, well, infectious diseases are a big concern and they can cause devastating effects to an industry that is worth $39 billion in the US and 100 billion euros in Europe. Clear examples of these are, for example, equine influenza outbreaks and also equine arthritis virus. So equine arthritis virus is a major cause of global concern because it can rapidly spread through respiratory or venereal routes and causes extensive outbreaks of abortion, neonatal death, and also respiratory disease. But the most unique characteristic of this virus is that it can cause persistent infection in what we so-call carrier stallions. So these carrier stallions can shed the virus in the semen for extended periods of time. And now, to date, the mechanisms that lead to this persistence are not well understood. These carrier stallions can, of course, transmit the virus through not only natural breeding, but also through use of contaminated semen and also using embryo transfer technologies. Today, there are no known therapeutics to prevent and to clear the virus infection from the reproductive tract of these stallions, and therefore, the only method available is to resort to surgical castration, which is a huge economic burden to the equine breeding industry. So in order to develop novel therapeutics, what we need to know is to further understand why persistent infection occurs in stallions and, well, what are the mechanisms involved in this. So my research is focused on understanding these and understanding how the stallion's immune response takes place during persistent infection. And we have demonstrated that the virus uses complex mechanisms to subvert the immune response and to evade the host immunity by infecting lymphocytes in the reproductive tract, and also by evading local inflammatory responses and also antibody responses in the seminal plasma. So now the key to elucidate the mechanisms that allow long-term persistence include a further analysis of the immune response in the reproductive tract and also linking this immune response to the host genetic background. Thank you very much.